Nos encontramos en el laboratorio de fitopatología.
We are in the phytopathology laboratory of the Las Lagunas Experimental Center, which is part of the National Rice Fund, FEDEROS. In this laboratory, we carry out some activities that have to do with the diagnosis of rice crop diseases. It is a service provided to the farmer. Here we can know precisely what pathogen is affecting the rice crop, as well as being able to have preventative measures for disease control in this crop.
El laboratorio de fitopatología.
The plant pathology laboratory fulfills several objectives, one of which is a diagnostic service to farmers, technical engineers, and all of those interested in cultivation. The diagnosis is mainly based on the application of techniques that allows to identify very clearly which pathogens are affecting the crop in order to issue a report in which the disease that is affecting the crop is listed, as well as the best control measures to be taken.
Campos arroceros. Otro objetivo muy importante.
Another very important objective of the phytopathology laboratory is to estimate the phytosanitary status of the different areas or regions of the country where rice is grown. In this way, the laboratory coordinates sampling to determine which of the diseases is most intense or present in the crops of each region, and thus be able to issue early warnings and or control measures.
Otro aspecto muy importante tiene que ver con.
Another important aspect is the monitoring of white leaf virus, which is a disease that affects all parts of the rice plant. Here we intend to determine the incidence of the disease in the rice fields, as well as the virulence status of the main sector of the white leaf virus. The other factor is the vector that is capable of transmitting the virus, which is the Sagata insect, so that we can determine each area what are the alerts and control measures for this important disease.
También hay la posibilidad de hacer estudios en este laboratorio.
There is also the possibility of carrying out studies in this laboratory related to viruses, pathogens such as fungi, bacteria, and some nematodes. We provide the service of microbiological analysis of the soil, where we determine the quantity and quality of each of the populations of microorganisms that make up the soil and that allow in some way to have the useful life of the crop of the soil. Rice in each of the regions and in each of the country's rice plots.
Para complementar como un diagnóstico o dar un.
In order to provide a more complete diagnosis, we carry out a process of identification of the type of bacteria by means of a SPG, which gives us purple colonies. We also carry out PCR tests, which is carried out by means of the DNA extraction method.
PCR, que es por el método de extracción de ADN. Bueno, el laboratorio también presta un servicio.
The laboratory provides a soil diagnostic service, also known as soil microbiology.
Diagnóstico, donde podemos decirle al agricultor con qué.
From this, we tell the farmer what type of microorganisms or nutrients his crop has.
La preparación de 8 medios de cultivos, prestándole las nutriciones que pueda tener.
Then, what we do is a presentation of 8 culture media, now by means of a guide.
¿Qué son los que qué microorganismos nos puede mostrar? Entonces, en base a ellos tenemos uno de los.
They will show us the necessary microorganisms and nutrients. Based on this, they give us a sign in the form of circles, that is, the crop is separated from the color that we previously added to it. Our function in this soil microbiology study is to tell the farmer how many colonies his crop has.
Eso es parte del diagnóstico que nosotros le entregamos como una microbiología de suelos.
Para diagnóstico, para la recepción de las muestras.
For the reception of the samples, the farmer must bring an entry form, or we take the respective data with the information of the lot and its municipality.
La muestra viene de lejos, entonces nos traen esta muestra empacadita, ya con la ficha de entrada.
When they bring us a sample from a distant territory, they must bring it packaged and with the entry form completed, filled out, Then we roll it up in kraft newspaper. The sample must contain a little of the remains of the soil so that the plant can resist the 2 or 3 days that the shipping process involves.
De esta zona de Saldaña.
If the plant comes from this area of Saladaña or Prado Purificación, where it is possible that it will arrive the same day, that is, it will arrive fresh. The farmer can pack the sample in this way. They bring it in a bag. For example, this plant has stains on its leaves. So this is an example of how you should bring or send the plant if you are near our laboratory.
En esta área funciona el laboratorio.
The grain quality laboratory of the experimental center operates in this area. La Lagunas de Ferreros, Fondo Nacional de Arroz. Here we can estimate all the variables that have to do with the industrial quality, with the appearance of the grain and with its culinary quality.
Estas variables envuelven mediciones como los contenidos.
These variables involve measurements such as amylose content, gelatinization temperature, and rheological profile, the latter allowing us to determine how the rice grain ends up in the cooking process.
Son granos que tienen la posibilidad.
Our consumers prefer those grains that grow. In general terms, we can also evaluate the appearance of the grain.
En términos generales, también podemos hacer la evaluación de la apariencia del grano, donde podemos determinar el largo y ancho del mismo y además
where we can determine the length and the width of the grain, as well as the transparency or translucency of the rice grain.
De otro lado, podemos establecer todos los parámetros del proceso industrial
in terms of a product that is pleasing to the eye. On the other hand, we can establish all the parameters of the industrial process, from the moment the green paddy grain enters until we have a white grain in the hulling, polishing, and classification processes, which allows us to establish the depulated index, which is the amount of grain that can be recovered whole from a sample of parent rice. This is how the laboratory allows us to establish all the parameters that future varieties may have to satisfy the industrial needs, the final consumer, and of course the Colombian rice farmer.
Para la evaluación de la calidad industrial del arroz.
I would like to tell you about the evaluation of the industrial quality of the rice. Once a sample of green paddy grain from the field that has been harvested at the right time, for example, with a grain moisture between 22 and 25%, we start the process of removing impurities and then drying the sample, for which we use a tower of warm air, which is around 35 to 38 degrees Celsius, by means of the flow, and a sample allows us to perform the drying. This to reach the point of 14% moisture, which is considered our dry paddy grain.
Encuentra con una humedad del 14%.
Once the dry paddy grain has a moisture content of 14%, we start the wheat process, which begins with the husking process, that is, removing the husks from the rice grain and giving us the first byproduct known as a whole grain.
En este proceso debemos retirarle entonces una capa de aleurona que trae el grano de arroz, donde concentra la mayor cantidad de nutrientes.
Using abrasive methods and a polisher, we will remove a layer of aileron from the rice grain, where most of the nutrients are concentrated in order to obtain the white grain or white mass.
Obtener el grano blanco o la masa blanca.
As far as the culinary quality of rice is concerned, one of the main characteristics has to do with the amylose content,
De cadenas,
which are fractions of trains that make up the starches in this case of rice. How do we determine it in this laboratory? Using a method, colorimetric method, which allows us to determine, by staining different types of rice, by appearance or color, the amount of amylose that a grain of rice may have. This amylose content is very important for the culinary quality of the rice, as it determines to a large extent whether the rice will end up with a loose cooking or a glutinous or sticky cooking. When amylose contents are high, these are generally rices that when cooked are loose, and when amylose contents are low, these rices generally end up as glutinous or sticky rices.
Generalmente estos arroces terminan siendo arroces glutinosos o pegajosos. En este caso, por ejemplo, tenemos variedades de amilosa alta.
In this case, we have varieties of high amylose, in which we see a blue staining that is determined in a spectrophotometry apparatus.
Los baja pueden teñir de esta manera. Sin embargo, estas muestras se llevan.
And some genotypes or varieties that may have low amylose usually stain in this way. However, these samples are taken to spectrophotometer to know the color of these, since it will determine the amount of amylose that each one of them contains. Another variable that we can measure in the gelatinization temperature, which also correlates strongly with the cooking of the rice. And we do that through alkaline digestion, which we do in these cuvettes. The grains that deteriorate faster in that digestion are going to allow for rice with lower gelatinization temperatures, which is what we are interested in for loosely cooked rice. And those that remain whole are generally with high gelatinization temperatures, which correlates with grains that end up in their sticky cooking.
Otra de las evaluaciones muy importantes dentro de la evaluación.
One of the important evaluations in terms of rice quality has to do with the measurement or determination of the viscosity profile of the rice flour in the cooking process.
Podemos contar aquí en el laboratorio con un determinador rápido de la viscosidad.
In that order of ideas, in the laboratory, we can have a quick result on the viscosity or an RBA equipment. This allows us with very small sample of rice flour to know its behavior during the cooking process.
Esa simulación va directamente relacionada con la temperatura.
In that process, we can simulate in the equipment that happens to the rice when it is being cooked in each of our homes. The simulation is directly related to the temperature that the rice undergoes during the cooking process. That is how this red line shows. This is how this red line shows us what is happening at the moment of cooking our rice. The rice starts in cold water, and we strongly increase the temperature until the water is boiling and a plateau is established in this process. When the rice loses supernatant water, it starts to be a slightly drier rice. The temperature in the cooker is lowered and the temperature starts to drop sharply.
Amigos de TvAgro, para nosotros ha sido un enorme placer.
Friends of TvAgro, it has been a pleasure for us to present a part of our work, which we carry out at the Las Lagunas Experimental Center as a contribution to the competitiveness and sustainability of our farmers and our country's rice sector. Thank you very much.